Okay, today we are going to learn about the role of myelination on action potential. At first, I want to mention that not all the neurons are myelinated. So there is a category of non-myelinated neurons and myelinated neurons. So we will see the difference in these two categories of neurons. The first, I want to mention that the velocity of action potential in non-myelinated neuron, example is multipolar interneuron, is approximate 0.5 to 2 meter per second. Now this is a very fast speed, but even such fast speeds are insufficient to permit the complex movements typical of animals. I want to give one example. In adult humans, there is a motor neuron that actually innervates the leg muscles. Now the cell body of this motor neuron is present in the spinal cord, and the length of the action is approximate 1 meter. Now suppose if the action potential velocity is 1 meter per second, then the action potential generated in the spinal cord will take 1 second, at least 1 second, to reach the leg muscles. Now for the coordinated muscle contractions required for walking and running, it is impossible if the action potential takes time approximate 1 second. So evolution has played an important role, and by this wrapping of myelinations, basically this is called myelin sheath, the velocity of action potential in myelinated neuron, example like motor neurons, increased approximately 10 to 120 meter per second. Now see the difference, it will approximate 20 times to 60 times, maximum 60 times of the action potential velocity. So basically the role of myelination or action potential is to increase in conduction's velocity of action potential. Now what is the reason behind this increase? The reason is myelination actually prevent passage of ions across the plasma membrane of actions. Now it prevents the movement of ions. Now in my previous video, the generation of action potential, I mentioned in the actions there is a voltage-gated sodium channels and potassium channels that actually through which the ions move and action potential is generated. Now if myelination prevents the passage of ions, now how the action potential is generated? Now if we see the microscopic structure of action, you will find that myelin is not wrapped all around the actions. There is a small gap approximately 1 micrometer that's called the nodes of Ranvier, where there is no myelin sheath. Okay, now the nodes of Ranvier is very important because nearly all the voltage-gated sodium channel, potassium channel and sodium potassium ATPase pump are concentrated here. So this is the site where action potential can be generated. Another site is present called action hillock, but these nodes of Ranvier can be the site where action potential could be generated. Now action potential, if generates any of the nodes of Ranvier, it actually jumps from one node to another node, and this type of movement is called saltatory conductions. Okay. Now sodium ions, basically what happens when the depolarization occurs, voltage-gated sodium channels actually opens, and through the voltage-gated sodium channel, sodium ions enter inside the actions. So sodium that enters through the nodes of Ranvier actually spread passively to the next node. So it spread through the next node. Okay. Now I have made a separate video on the directions of action potential. In that video I mentioned that action potential always move away where from it is generated. Okay, suppose here the action potential is generated, so it will move away, it will not move in this backward directions. What is the reason? In that video I have mentioned that due to the inactivation of voltage-gated sodium channel. So sodium channel has an intrinsic property, it remains closed during resting period of the neurons, then when depolarization occurs it becomes open and quickly it becomes inactive. Now this voltage-gated sodium channels remain inactive for approximately 4 milliseconds. This is called a refractory period. So in that time period the voltage-gated sodium channel could not be open again. So the sodium enters through this segment of the nodes of Ranvier. Suppose here the action potential generated, it will spread here and it will activate or open the voltage-gated sodium channels present in these nodes. So what happens? Another action potential will be generated in this segment. Now the sodium that enters through this segment will passively contact here and in this direction. But as because this sodium channel becomes very quickly inactivated, they could not be opened again. So action potential actually move always away from the initial generation site. Now two types of glia or glial cells actually makes the myelin sheath. One thing I want to mention that myelin sheath could be multiple layered. Okay. One example of glial cell is called oligodendrocytes. These cells present in the central nervous system, brain and spinal cord. So the neurons that present in central nervous systems actually wrapped by oligodendrocytes, and it wrapped multiple neurons. So one oligodendrocytes can wrap multiple neurons. Two important, this is the major proteins present in these cells, or actually the myelin sheath, is the myelin basic protein, in short MBP, and proteolipid protein PLP. The second type of glial cells, that is called Schwann cells, it is present in the peripheral nervous system. So nerves or neurons that is present in the peripheral will be wrapped by Schwann cells, and this type of cells actually wrap only one neuron. So there is a difference. It can only wrap a single neuron. Now there is a major protein called protein 0, or in short P0, and it is present approximately 80%. Okay, now let me talk about some demyelinating disease. That means if there is any damage occur to the myelin sheath, this type of disease can be manifested. The one example I have taken, that is multiple sclerosis, in short we call MS, and it causes neurological disease. Now it is very simple to understand that, as because myelin sheath is present in the neurons, so if there is any damage occurring in the myelin sheath, that means there will be some neurological diseases. Now this disease is basically characterized by spasms and weakness in one or more limbs, also bladder dysfunction, local sensory losses, visual disturbances occur. Now all these symptoms occur due to the patchy loss of myelin present in the brain and spinal cord, basically the neurons present in the brain and spinal cord. And why these symptoms occur? Because the conduction of action potential is actually slowed. As I mentioned, that myelination actually increases the velocity. So if there is any problem occurring in the myelinations, then the action potential conduction is actually slowed. Now the reason or the cause of this disease is not known completely, so it is unknown, but it appears that there is a generation of autoantibody against a protein called MBP that I have earlier mentioned. MBP means myelin basic protein. One very interesting thing occurs in case of mouse mutant called severer. Here also a deletion of much of the MBP, that means myelin basic protein gene, occurs. They also show neurological symptoms like tremors, convulsions, and early death. Pelagius marked by the disease in case of human, and GMP in case of mice. So in both these situations, mutations occur in the PLP genes, that means proteolipid protein gene. And I have mentioned earlier that in case of central nervous systems, oligodendrocytes or glial cells actually makes this myelin sheath, and basically these two diseases occur due to the damage of myelin present in the central nervous system neurons. Now the third is called Guillain-Barré syndrome. Now here the damage occurs in the peripheral nervous system, so peripheral myelin is targeted. This is also due to it is assumed that it is due to the autoimmune reaction. Now the cause is not completely known, so it is unknown, and generally the symptom is rapid onset of paralysis occur.